Every type of virus. RNA virus. These viruses use RNA as their genetic material instead of DNA, and that single difference changes everything about how they behave inside the body. RNA is not as stable as DNA, so when these viruses make copies of themselves, the copying process is more error-prone. That means every time they reproduce inside a host cell, small mistakes are introduced into their genetic code. At first, those mistakes might not seem important, but over time they build up and create completely new versions of the virus. This constant change is what makes RNA viruses so unpredictable. In real life, this is one of the main reasons why certain illnesses, especially respiratory infections like influenza, seem to return again and again in different forms. Even if your immune system has already learned to fight one version, a slightly changed version can still infect you. This ability to rapidly evolve allows RNA viruses to adapt to new environments, new hosts, and even immune defenses, which is why they are constantly monitored in public health systems. DNA virus. DNA viruses are different because they use DNA as their genetic material, which is much more stable and accurate when it is copied. This stability means they do not change as quickly as RNA viruses, and their structure often remains more consistent over long periods of time. When a DNA virus enters the body, it attaches to host cells and begins its replication process in a more controlled and slower way. Because of this, the immune system can sometimes recognize them more effectively if it has encountered them before. However, this does not make them harmless or easy to eliminate. One of the most important features of DNA viruses is their ability to remain inside the body for long periods without showing clear symptoms. In some cases, they become inactive and hide inside cells, almost like they are sleeping. Later on, they can reactivate when the immune system is weakened or when the body is under stress. This combination of stability and hidden survival makes them persistent and sometimes difficult to fully clear. Retrovirus. Retroviruses are unique because they do not follow the normal biological flow of genetic information. Instead of simply using their RNA to make copies of themselves, they first convert their RNA into DNA after entering a host cell. This process is called reverse transcription. Once that DNA is created, it is inserted directly into the host cell's own genetic material inside the nucleus. At that point, the virus is no longer just an external invader. It becomes part of the cell's instructions. Every time the cell divides, it also copies the viral DNA along with its own. This means the virus can persist for a very long time without being eliminated. Because it is hidden within the host's genetic code, the immune system has a very hard time detecting and removing it completely. This deep integration is what makes retroviruses especially long-lasting and complex in the way they affect the body. Enveloped virus. Enveloped viruses are surrounded by a soft outer layer that they take from the host cell when they leave it. This outer layer, called an envelope, is made mostly of lipids, which are similar to fats. It plays a very important role in helping the virus enter new cells. The envelope allows the virus to merge with the membrane of a healthy cell, almost like two bubbles fusing together, so the viral material can enter smoothly. However, this same structure also makes them fragile outside the body. Because the envelope is soft and delicate, it can be easily broken down by soap, alcohol, heat, or drying conditions. Once the envelope is damaged, the virus becomes inactive and can no longer infect cells. This is why hygiene and cleaning are very effective against them. Even though they are fragile in the environment, inside the body they can be very efficient at spreading because of how easily they enter cells. Non-enveloped virus. Non-enveloped viruses are more resistant because they do not have a soft outer membrane. Instead, they are protected by a strong protein shell known as a capsid. This capsid acts like armor, shielding the virus from harsh environmental conditions. Because of this protection, non-enveloped viruses can survive for much longer periods on surfaces such as phones, desks, door handles, or other frequently touched objects. They are also more resistant to drying, heat, and some cleaning conditions compared to enveloped viruses. This makes indirect transmission more likely, since a person does not always need to meet an infected individual directly to become exposed. Simply touching a contaminated surface and then touching the face or mouth can sometimes be enough for infection to occur. Their durability is one of the main reasons they can spread effectively in crowded or shared environments. Airborne virus. Airborne viruses spread through extremely small particles that are released into the air when an infected person breathes, talks, coughs, or sneezes. These particles are so small that they can remain suspended in the air for a period of time instead of falling immediately to the ground. Because of this, infection can happen even without direct contact with an infected person. A person may simply breathe in the particles without realizing it. The way air moves in a room becomes very important in how these viruses spread. In closed or poorly ventilated spaces, particles can build up and remain in the air longer, increasing the chance of infection. In contrast, open spaces or good ventilation help dilute and remove these particles, lowering the risk. This invisible nature of transmission is what makes airborne viruses particularly important to understand. Zoonotic virus. Zoonotic viruses are viruses that originate in animals before making the transition into humans. This process usually begins when humans come into close contact with infected animals, their fluids, or environments where the virus is present. Sometimes it can happen through handling animals, sometimes through bites, and in other cases through contaminated food or water. In certain situations, insects can also act as carriers between animals and humans. Once the virus successfully adapts to human biology, it may begin spreading from person to person without needing the original animal host anymore. This transition is not immediate. It often involves small genetic changes that allow the virus to survive in a new species. Many significant outbreaks throughout history have started in this way, showing how interconnected human health is with animal populations and ecosystems. Vector-borne virus. Vector-borne viruses depend on another living organism, usually an insect, to carry them from one host to another. Mosquitoes are the most common example, but ticks and other biting insects can also act as vectors. The virus does not usually harm the insect itself. Instead, it uses it as a transportation system to reach a new host. When the insect feeds on blood, the virus is transferred directly into the bloodstream of the next organism. Because of this, the spread of vector-borne viruses is strongly influenced by environmental conditions such as temperature, rainfall, and habitat. Warmer climates often increase insect populations, which can increase transmission rates. This makes vector-borne viruses closely linked to geography and climate patterns rather than just human contact alone. Latent virus. Latent viruses have the ability to remain inside the body in a hidden, inactive state after the initial infection. During this period, they do not produce symptoms and may remain completely unnoticed for long periods of time. Instead of being fully eliminated by the immune system, they stay inside certain cells in a dormant condition. This is almost like the virus is paused but not gone. Later on, if the immune system becomes weakened, or if the body goes through stress or other triggering conditions, the virus can reactivate. When it becomes active again, symptoms may return even though the original infection seemed to have ended. This cycle of hiding and reappearing makes latent viruses particularly persistent and difficult to fully remove from the body.